microbial diversity, disease diagnosis, human and animal genetics, medicines, and antibiotics development. So metagenomics could be used to study the human microbiome, which is like gut microbiota, skin microbiota, and their roles in health and disease. It can be used to identify the pathogens in clinical samples, which helps to diagnose and treat infectious diseases. Metagenomics helps in understanding of microbial diversity and functions of different soils in different geographical locations. It can be used to monitor water quality and detect pollution by studying microbial communities in different water samples. It can also be used for in bioremediation efforts by identifying microbes that are capable of degrading the pollutants. It, understanding the microbial communities associated with plants can improve crop health and productivity. It helps in identifying microorganisms that are capable of energy production, which is useful for the production of biofuels and other renewable energy sources. It is useful in discovering new enzymes, biotic compounds, antibiotics, pharmaceuticals, and other beneficial microbial products. If you are not interested in reading such detailed texts, then this right side graphical representation is more than sufficient to understand the application of metagenomics. Now, we can summarize how it can help. It can help in microbial studies, disease biomarker discovery, antibiotic resistance tracking, and personalized therapeutics approach, for example, in cancer, diabetes, and some other diseases. Now, it is used for soil health monitoring, plant microbe interaction studies, crop yield improvement, pest and disease resistance understanding. Now, the application, as I mentioned, in the sense of now different fields of application, if we classify, then in terms of environmental science, it is helpful in pollution monitoring, biogeochemical cycle analysis, climate change impact and studies, bioremediation. Then, in terms of biotechnology, it helps to identify enzymes in synthetic biology, biofuel production, and in industrial microbiology. Now, it is also used in marine biology, that is ocean microbiome mapping, coral reef health assessment, carbon cycling studies, marine ecosystem monitoring. Now, if we are going to look for the limitations of metagenomics, then there are a number of limitations. For example, cloning biases can lead to misrepresentation of microbial DNA. Sampling biases can occur when collecting environmental samples, can also lead to misrepresentation. This sequencing can detect the presence of microbes, but cannot easily determine if they are pathogenic or non-pathogenic. The vast amount of metagenomic data requires faster and more scalable computational tools. The data sets contain a mix of DNA from various organisms, which often makes difficult to assemble genomes more accurately. Contamination from host DNA or environmental sources can affect accuracy of metagenomic analysis. It is also challenging to extract genes from microbes present in a low abundance within a sample. So, examples of metagenomics projects. These are the big projects which I have included here as an example. One is Human Microbiome Project, which is one of the important examples of metagenomic projects. It was launched by the National Institute of Health, NIH, in 2007 to study microbial communities present in different parts of human body, such as gut, skin, and mouth. Global Ocean Sampling Expedition, that is GOS. This project aimed to study microbial diversities present in the oceans using shotgun metagenomics sequencing techniques. Then it is IMG that is integrated microbial genomes and microbiome system. This is a database platform that focuses on microbial genomes and microbiomes. It provides access to metagenomic data and also provides tools for comparative analysis of microbial genomes. So if you want to keep it in track, you can subscribe the channel, which includes the complete lectures in different parts. So it is always easier to subscribe and get the updates when we are uploading the new lectures and videos regarding any topics in biological sciences. Now, I'm going to summarize the methodology. Methodologies in general, it is just sample collection, DNA extraction, sequencing, quality control, assembly, and binning, annotation, and analysis. Then the steps involved, for example, first step is sample collection and processing. So all those people who are experienced in this field, they know how to collect the samples and how to process it, and then go for metagenomic library preparation, screening of metagenome library. Then fourth step is DNA sequencing, sequence assembly, then binning, which is basically the part and process of grouping or sorting assembled DNA sequences into groups or bins that represent individual genomes from closely related organisms. Then seventh step, let's say annotation. Eighth step is experimental design and statistical analysis. Finally, to store all the data and share it with publicly accessible databases such as SRA, NCBI, SRA. So these are some of the selected steps, and the details are mentioned in the text. You could also look over these texts. Now, if we want to summarize these steps as a command pipeline with an example, and the easiest pipeline with the software names, command names are Velvet G. So for example, here I have summarized. Use Velvet G, use Diamond software, then use Migan software. It is quite easy and fastest way to analyze such complex data in just few days. So first part, remember it, Velvet G, and then Diamond, then upload the Diamond meganized files. So